Welcome, dear students. Today we're going to start and to continue the chapter 5, DNA and genetics, starting with the difference between the DNA in eukaryotic and prokaryotic. Let's start the lesson. Enzymes and DNA replication. In the last episode, we studied how the enzymes affect the DNA replication or duplication. And right now, we're going to continue the process of replication of DNA. And we have here a picture to show that the different types of enzymes like polymerase and ligase, as we studied them. As we said, we have polymerase, helicase, and each one of them has its own function. One of them to separate the two strands of DNA, and one of them to get the missed nucleotide to make the template or the complementary side of the DNA. After that, we need to get some information about the DNA in eukaryotic and prokaryotic. The DNA of eukaryotes are the living organisms. Their nuclei, they are surrounded by nuclear envelope, okay, is organized into several chromosomes. There are a lot of chromosomes, and each one of them has its own function. Each chromosome contains a single DNA molecule, which runs from one end of the chromosome to the other end. Also, the replication starts at many different points along the molecules, okay? It doesn't necessarily to start from a definite or a specific point. While in case of prokaryotes, the DNA, it exists as a double helix. With its ends, they are joined to form a kind of circle. They looks like as a circle. But in the eukaryotes, the DNA, it exists in form of chromosomes, not circle as in prokaryotes. The molecule also is attached to the plasma membrane at one point. This point where the replication takes place or occurs in case of prokaryotic. DNA repair, this is the much more information that we need to know it about the DNA repairing, how the DNA recovered or repairing if a part is damaged along the double helix of DNA. Actually, all biological polymers, long molecules, they are formed of repeating units such as starch, proteins, nucleic acid, enzymes, whatever, any macro and micromolecules, as you studied in the first secondary. They actually containing a kind of repetition of units, okay? Well, they are subjected to damage from the body itself by heat and fever, high temperature, low temperature, from different environments, aqueous, solution, aqueous or salty bases, and so on. All these circumstances or these aqueous environments inside the cell, DNA, there's no exceptions, and also DNA, and the function of the DNA to repair, okay, and to recover the missed and the damaged macro and micromolecules inside our body. It was estimated or appreciated that 5,000 parent pieces and guanine are lost each day from the DNA of each human cell. So we have a great number of missed and damaged and destroyed pieces each day. So we need a kind of a mechanism or a property inside our body to prepare the destroyed and missed one. Let's know it. Because also the heat, it has an important role in breaking down the covalent bonds that link them or link the nucleotide together, them to the oxyribose. DNA also can be damaged by a different ways, like a various chemical compounds and radiation, heating, whatever. I mean that the main point here that we have a lot of circumstances, a lot of reasons they are caused damaging and destroying the DNA. Okay, like temperature, like a different chemical environment, like a different radiation. We every day we subjected and exposed to a lot of these factors, and they are all of them they are destroyed and cause damage to the DNA. But how can we repair it? The question here is how the DNA repair itself. Any form of damaging to the DNA could alter mean we can use it, we can change it, its information content. Again, any damaging or destroying that occurs or takes place to the DNA, it could alter or change, okay, the information content. Seriously, yes. So, did you mean that by causing the damaging of DNA, that one of the greatest reasons of occurring or happening the mutation or appearing of undesirable factor or undesirable character in the living organism? Yes. So that may produce disastrous change or kind of malformation or deformation in the cells proteins. Although the thousands of changes occur in a DNA molecule every day, but the DNA itself, it is still in constant and fixed way. I mean, it contains a kind of stability. No more than 2 or 3 stable changes accumulate in a cell's DNA each year. The vast majority of the changes are eliminated with a remarkable efficiency by a group of 20 different kinds. And actually, I think this is one of the main reasons of the stability of the DNA within and inside your body. DNA repair enzymes. As we have got some enzymes to replicate and decompose the units or nucleotides from which the DNA are formed. We have also some enzymes, okay, to repair the missed part or damaged or destroyed part of the DNA, as we said, by different factors or different criteria and circumstances, like heat, like temperature, like chemical, like radiation, and so on. From these enzymes, such as what? Actually, these enzymes, which are called DNA ligases, this group of enzymes, they work together in harmony, okay? There is a kind of synchronization between them, and they can recognize and remove the damaged area, the damaged piece or part of DNA, and rapidly they repair it. So we have a group of enzymes, they are called DNA ligases. This family of enzymes, they are working together in harmony, okay? They synchronize together, pair together, to remove the damaged area, and at the moment they rapidly repair the damaged part of the DNA. Were they replacing the damaged part with the nucleotides, with the complementary piece or parts of it, to those of the other strand or the opposite strand to the damaged portion. For example, if you have a piece of the DNA which includes on the right side adenine, okay, and followed by guanine, and on the other strand or the other side you have what? You have cytosine followed by cytosine or the complementary to it. If this part is absolutely damaged, exposed to total damage, the enzymes or ligases, they have ability to repair and to bring the missed part of the DNA to mismatching nucleotides. DNA also repair depends on the existence of 2 copies of the genetic information. One of these copies in each strand of the double helix. As long as one of these strands remains undamaged, we have a condition here. If the one of these strands is still as it is fixed or constant, there is no damaging in this strand, what's happening? The repair enzymes can use it as a template. It can use it as a complementary site, okay, to complete, to fill the missed nucleotides from it, to replace the damaged segment or part or piece in its partner on the other side. But we have a condition here, as we said, as long as the other side of the template or the other complementary backbone or side or ladder of the DNA helix, it is still as it is without damaging. So most damaging is it can be recovered, okay, unless both strands are absolutely altered. They're totally changed, altered or changed at the same location. I mean the total part, okay, of the double helix DNA, okay, in the both sides are completely damaged, missed, okay, the same location at the same time. That may causes the mutation or the change in the content of the DNA itself. The genetic material of some viruses occurs and takes place in the form of a single strand RNA. So, as we studied the bacteriophage, if you remember that, as we said, that bacteriophage is a type of viruses, okay, and it contains DNA, DNA molecule or material, genetic material, and coated by a protein coat. But some of the viruses, they don't have the DNA, but they have what? RNA, ribonucleic acid. I was going to study it, but which cannot be repaired. The RNA of the viruses, it cannot be repaired. So these viruses show high rates of genetic changes and resulting from damaging to their RNA. And this type of virus, actually, it's a very difficult to remove it from our bodies, and also it's a very difficult because they are a lot of we don't have the fixed form or fixed chip for the sequence of their DNA, okay, to eliminate it, remove it from our body, okay, but they have a lot of changes, okay, because the DNA itself, RNA itself, sorry, it doesn't repaired easily as the DNA, okay. I mean the missed part, we cannot repair it. The double helix, the vital to the genetic stability of the organism that contain it, as we said. Well, DNA in prokaryotes, on the other hand of the eukaryotes, as we studied, it has ability to be repaired and also to be restored and recovered in most of the most of the living organisms, except some types of viruses, which includes RNA, not DNA. The genetic material of prokaryotes is one of double helix of DNA, with its ends, as we said, joined to each other, forming what? A circle. They aren't exist in form of chromosomes. Okay, this is one of the main differences between the DNA form in the both of prokaryotes and eukaryotic. if the DNA of the bacterium, I mean it's a type of bacterium which is called Escherichia coli, and this type of bacterium, it lives in the colon, okay, definitely in the large intestine, okay, in the last part of the large intestine, where they are stretched out. I mean, if we bring DNA of this bacterium or this Escherichia coli, and we're going to stretch it, make it unwounded, okay. I mean, we unwound the double helix to be a straight line. It would be about 1.4 millimeter long. And is it a long? Sure. Because if you know that the cell itself only it's about 2 micron long, do you believe that? So the double helix and twisting of the DNA, it's a very tightly and highly tighted inside the nucleus, and the nucleus inside the cell itself. Believe that when we go into strain the just one living organism which is a unicellular, about 1.4 millimeter, where the cell itself it measured by 2 micron long or less than that. The circular bacterial DNA is folded many times and occupies a nuclear area, about 1/10 of the cell's volume. I mean 1/10 from the total volume of the cell. Yet the DNA molecule is attached to plasma membrane at one end or more points. I mean, it sometimes connected to the plasma membrane just by one end or more than one point. Also, in addition to that, some bacteria also they are contain one or several additional or much smaller circular DNA molecules. They are called plasmids. Again, plasmids, they are what? They are several or smaller circular DNA molecules called plasmids. Plasmids actually are widely used in the genetic engineering. This field of the science, it depends on the information and how can we control the genes or the information data on the chromosomes, okay, called genetic engineering, as will be discussed inshallah in the next lesson or the last chapter. Bacterial cell also replicates. Any plasmid, it contains at the same time as its main DNA replicate. So scientists take this advantage and they meet a kind of the activity by introducing artificial plasmids into bacteria. For what? Why they did this experiment? I mean, they are forced or made the kind of introduce the artificial plasmid into a bacteria to obtain several copies of these bacteria. Okay, and we have here the DNA in prokaryotes. Okay, but actually it's magnified by electron microscope, or we have here the electron microscope showing the DNA in prokaryotes or the bacterial cell. Okay, the DNA molecules also of mitochondria and chloroplasts, I mean the organelles of eukaryote itself, they are very similar to that of the prokaryote. So we have here a kind of similarity. If I'm going to ask about the similarities between both of DNA in prokaryotes and eukaryotes, you have to tell me this information, that the DNA of molecules, mitochondria and chloroplasts, I mean they are organelles. Both of them, they are exist in pro and eukaryotic cells. They are similar to each other. The DNA of these organelles Organelle, it's mean a tiny organ, okay? This mean of the organelles. And the yeast plasmids, they are circular and they aren't complexes with the proteins. I mean, they aren't combined or connected with much more proteins. So they are always found with eukaryotic DNA. The structure of eukaryotic cells. The chromosomes, starting with the chromosomes. Chromosomes appear in eukaryotic cells during cell division. Let me to remind you the cell division here. Cell division, as we said, when the one cell, for example, the mitotic division, such as what happen in every day inside the somatic cells or the body cells. We need the one cell to be 2 and 2 to be 4, okay? And this is type of the cell division. The chromosomes or the condensation, the intensing or intensifying of the genetic materials appears in the X shapes or the crosses of chromosomes appear in this type, in the form of chromosomes during what? During the cell division. Each chromosome is thought to contain a single DNA molecule extending from one end of the chromosomes to the other end, but coiled and folded many times. The DNA also is associated with the various proteins. DNA is associated or connected, linked with different types and various types of proteins. Each one of them has its own function. Forming what? They form something called chromatin. So we have the chromosomes, they appear during the cell division, and after that they are combined with a various of proteins. Each one of them has its own function. They form something called chromatin, or chromatin kind of the other type. We have two different chromatin reticulum, which contains a roughly equal amount of DNA and proteins. So the chromosomal proteins may be divided into histones and non-histones proteins. So again, we have two types of the chromosomal proteins. One of them is histone 1, and the other type, which is non-histone proteins. Histones make up a well-defined group of structural proteins, okay? And also histones are, all of them are very small. And they have a high content of the basic amino acids, okay, like lysine. At the pH inside the cell, pH here it's mean what? The concentration of the hydrogen, okay? pH of something like, for example, the pH of water is 7, so it's a neutral, okay? pH of acids, it's less than 7, okay? About 6, 5, 4, 3, 2, 1, and by decreasing the pH value, okay, the acidity increases, and by increasing the value of pH over 7, the medium gonna be what? Gonna be a basic medium. Okay, so we have three different mediums here: acidic, neutral, and basic one, or alkaline one. In case of the amino acids of the pH inside the cell, the amino acids, from its name, acid, so it should be what? Less than 7. They have a positively charged R groups or alkyl groups, as you studied in the chemistry. Or alkyl groups is maybe what? CH3, C2, and C2H5, and so on. Kind of alkyl group, okay? Any alkyl group. And so they are bounded strongly to the negatively charged phosphate groups of DNA. Histones occur in not several, but in enormous amount in the chromatin of any cell. The non-histone proteins also are heterogeneous. Heterogeneous means they aren't the same. They aren't the same. They are different from each other. Okay, they are contradictory to each other. Heterogeneous group with many functions. Each one of them has different functions. Okay. They also include some structural proteins, which play an important role in what in the spatial organization of the DNA within the nucleus, and also they include the regulatory proteins, and we're going to get some information about the regulatory proteins, or protein that has important role in the regulation, which determine or not the DNA code is used to make RNA. Okay, and the next chapter we're going to study the difference between DNA and RNA coding and uncoding. So and also it contains what whether or not the DNA code, and also to make RNA, also to make proteins and enzymes. The human somatic cells contains 46 chromosomes. Each somatic cells, each cell inside your body, it contains definitely 46 chromosomes. Each one of them, it represents and reflects one of your character, one of your feature, physiological, psychological, whatever, but it already exists. If we imagine that the DNA double helix from these chromosomes were lined up, I mean, it's gonna be linear. Okay, we make it as a straight line, and they stretch it out. They would be about 2 meters long. Yes, and imagine that the diameter of the cell itself from maximum 3 microns. Okay, and while the DNA itself, if we're going to stretch it, we can measure it by meters. So the histone and the other proteins are responsible for packing and intensifying, condensing these long molecules into a nucleus. Okay, inside the nucleus, inside the cell itself, from 2 to 3 micrometer in diameter. At the end of the episode, I hope you get the difference between the eukaryotic and the prokaryotic DNA and how they are affect on the both structures of prokaryotic and eukaryotic, and the experiments that are carried out by the scientists. See you the next time, and goodbye.